This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 19 for February 7th, 2009. From somewhere in New Jersey, I'm Vincent Racaniello.
And from somewhere in western Massachusetts, I'm Alan Dove.
It's a Saturday. Alan is always lost.
Well, I like to tell people I'm in the Springfield area and then not tell them what state.
Springfield, Missouri, I think, right?
Springfield, Illinois. I think they're the story I heard was there's a Springfield in every state except Hawaii.
Well, did you have a great week, Alan?
I had a mediocre week. I had some kind of GI virus, I think, earlier in the week, and then took a little while to get over that.
Damned viruses.
Damned viruses, yeah.
Did you have a cytokine storm in your GI tract? Was that the problem?
That's what it felt like, yes.
I was listening to the infectious disease Pusscast the other day, and he started by saying he had a cytokine or an interferon storm in his upper respiratory tract, which I thought was good. I like that because, as we all know, the storm is part of the problem.
Exactly.
Well, we have a couple of interesting stories for today and some reader email, so let's get on the case here. Yesterday was the last day of TED.
Right.
TED.com, and our buddy Dick Despommier, of course, gave his talk yesterday, and I don't know how it went, but we'll hear when he gets back. They haven't posted the video yet.
So we'll have to find out if Dick was in the room when Bill Gates released more bugs into the world.
Exactly. We have to get the story right from Dick. I'm sure he's going to have a great story. So as everyone may have heard, Bill Gates, who spoke on Wednesday, released some mosquitoes into the audience. He had a jar, I suppose, up on the lectern, and he opened it up. And he said, not only poor people should experience this. And this was all over the place on Wednesday and Thursday. You must have seen it too, right, Alan?
Oh, yeah. It's... insert Windows Vista joke here.
He said they were free of malaria, and I asked on Twitter whether they were free of viruses.
It's a valid question. And one of the things I was wondering was, did he release Anopheles mosquitoes, which would be the malaria vector, or did he release something else? I mean, were these just tiger mosquitoes, or what? There are several species of mosquitoes.
Good point. Yeah, I don't know. We'll have to ask Dick if he knows. You would guess that he bought these mosquitoes at a biology supply place, right?
Probably from a lab supplier, yeah. So they would have been bred in captivity. And just to give a little insight on this, my my grandfather used to do this for a living. He was an entomologist, and they would raise biting insects of various sorts in the lab, and I don't know if the technique for doing this has improved at all, but at that time the only way you could feed them was to stick an arm into the cage. So volunteers they were called volunteers, would have the job of putting a can of lice on their arm, or putting their arm into a mosquito cage or what have you, and feeding the mosquitoes. So it seems pretty likely that these were free of any kind of pathogen you would need to worry about, because if they weren't, then the people who were responsible for feeding them would have come down with it. Unless there's some better way of doing it. I mean, I don't know. Maybe now they've got some better technique for feeding the mosquitoes, so you don't actually have to stick your arm in there.
Yeah, I just thought it was interesting that he made a point of saying they didn't have malaria, but he didn't mention anything else, right?
Right.
So just because we talk about mosquito-borne viruses a lot, he should have said probably there are no known pathogens in these mosquitoes, not just malaria.
Yes, that would have been a much better way to put it.
But his talk is up, and I started listening to it earlier. They're quite good, as they all are. So
right.
I think they will slowly put up the talks one by one, and we'll get to Dick's probably later, because he was the last, towards the end of the week.
Well, and it's complicated to put up a presentation like that. I do a lot of work with the New York Academy of Sciences, which actually does something very similar. They put a lot of their meetings online. So I know there's a lot that goes into, it's not quite as simple as posting a YouTube video.
I noticed that WHO has a web page on the cumulative H5N1 infections, and we'll put a link to that. But it's kind of an interesting table to look at. It goes from 2003 to the present, And these are laboratory-confirmed cases and deaths. So you probably will have heard of more H5N1 infections in the press than are on this page. They're only included here if they're lab confirmed. So from 2003 to the present, there are a total of 404 cases of H5N1 influenza in humans, 254 deaths. They're scattered all over. The main countries are Indonesia, Vietnam, China, Egypt, and then Thailand. Those are the main ones. And then a few countries have less. In 2009 so far, we have 9 cases and 4 deaths, which may be, again, less than what you've heard of in the press. 7 cases, 4 deaths in China, 2 cases with no deaths in Egypt. Considering how many people are in these regions, it's not very much. And clearly it hasn't been able to transmit yet among humans. Otherwise,
Right.
we would have far more cases, right? I saw a photograph this week in one newspaper. I don't recall where it was. It was a Chinese chicken farmer. He was sleeping in a room full of chickens. He was going to bring them to market the next morning, so I guess he had to sleep with them. And so can you imagine the aerosol of virus in that room?
If the chickens are infected, the farmer is infected, it would be fair to say.
Yeah, I mean, that's why many of these cases originate, because these people are working and sleeping right next to the chickens. So
Yeah.
It's not surprising if there's a virus in there. I would venture to say that if you went and trapped a few hundred mice from the wild and put them in a room and you slept with them, that you would probably get infected with some kind of virus also.
Quite possibly.
It's not surprising that this is happening, and with the increasing numbers in the chicken farming business, this is why this is happening more and more. Surprising at all. Anyway, we'll put a link to that. It's kind of a neat table.
Yeah, it is, and I presume they're going to be update. They don't they don't indicate on the page quite. It's got a date on the table of 27 January.
Yeah, if you back up one page on the crumb trail there at the top,
Got it.
you will go to a page where they have. It looks like every week they update it, yeah. So the most recent now is February 2nd.
Got it. Okay.
A lot of good resources out there. We talked last time about the role of adenovirus type 36 in obesity, and I had sent an email to my colleague at Columbia, Rudy Leibel, who's an expert in obesity. He was one of the people who had originally discovered leptin. And I asked him what he thought of this, and he wrote back, quote, "I think it unlikely that much, if any, obesity results from increased fat cell production. Still have to fill those cells," end quote.
Yes.
So yes, when you gain a lot of weight, the fat cells fill up. When you lose weight, they empty out, but the cells are still there.
Yep.
He doesn't think that induced proliferation in humans, at least, would play much of a role. So I guess we all have a lot of fat cells. It's just a matter of whether we fill them up or not.
Yeah, I've in covering a couple of conferences about obesity, one of the things that comes up is that you have this weight set point that gets established, and it's kind of established during puberty, and then it sort of gets reset periodically, and it's interesting that the number of fat cells you have seems like kind of a minor component of the weight set point. You may have a lot of adipocytes, but what matters is how much fat you're putting in them.
So I don't know. I mean, Rudy's an expert. I take his word for it.
Right.
But we'll see more on this because the story's not done.
Yeah, I think a lot of people, certainly people who become obese as adults, are probably not adding fat cells.
Right.
That's not the primary problem. The primary problem is that they're loading up the ones that they have.
Now, there were a couple of papers. I noticed a few this week. I'm not sure if they came out this week or not. There was they are both on hantavirus and on a particular protein of hantavirus is the N protein, which is an internal virion protein. These, of course, are enveloped RNA viruses. They're negative stranded, and their genome is coded with a protein, the N protein. And there were 2 papers on the function of this protein, which are very interesting. And the first one I came across is in the EMBO journal, called "A Protein That Replaces the Entire Cellular EIF4F Complex." So the EIF4F complex is a translation complex made up of a number of proteins that binds to the cap structure on mRNAs and basically allows the ribosomes to come in and start translating. And what they found in this paper was that the N protein of hantaviruses binds the cap with high affinity and basically displaces the EIF4F complex of the cell, which is very interesting because, when viruses infect cells, one of the things that happens in a defensive way is that the cell shuts off translation. And so the authors are speculating that maybe this is the virus's way of getting around that. The N protein goes to bind the caps, and then the N protein apparently can recruit ribosomes on its own to the mRNA.
Now, could this also be a way for the virus to preferentially ensure the translation of its own messages over the cell messages if translation is still occurring?
I would think so, yeah. I would suspect that so first of all, if it had a very high affinity, higher than EIF4F, it could displace cellular translation, right?
Right.
And then if the virus did inhibit cellular translation, then this would be a way of getting around that. So, Picorna-infected cells, EIF4G, which is a component of this 4F complex, is cleaved, and the virus gets around that by not even needing a cap. Its messenger RNA isn't capped, and it has an internal ribosome entry sequence instead. So yeah, you're right. It could be. They don't actually deal with that at all in this paper, but I do think that's a good point. It may have some role in selective translation of viral mRNAs. Then a few days later, I noticed a second paper on this same hantavirus N protein. And this was in PNAS. This, I believe, was in December. It's called Storage of Cellular 5' mRNA Caps in P-Bodies for Viral Cap-snatching. So these authors also find that the hantavirus N protein binds caps with high affinity, but they find that it binds... So the influenza virion polymerase requires primers in order to make mRNAs from its RNAs. And the primers are actually capped oligonucleotides derived from host mRNAs. The virions have endonucleases that cleave the mRNAs of the cell and use the resulting capped oligos to prime viral mRNA synthesis. Really neat process. And apparently those capped primers are bound by the N protein and stored in these places in the cell called P-bodies. P-bodies are depots for mRNAs that aren't being used at the moment. Okay, the cell seems to store them there, and then if they don't need them, they're sent somewhere to be degraded, but if they're needed, then they could be put back into the translation pathway. So apparently the N sequesters these capped primers in the P-bodies till the virus needs them. Very cool.
Now, P-bodies are a normal part of cellular metabolism in uninfected cells too, aren't they?
Absolutely, yes. They're in uninfected cells. It's thought that the cell, puts mRNAs there when it doesn't need to translate them at whatever time, because not every mRNA in the cell needs to be translated all the time, right? So it stores them in P-bodies and then pulls them out, calls for them when it's needed. And so as usual, the virus is usurping this pathway. It's storing its own primers in there. So this group shows that the N protein co-localizes with P-bodies, it's bound to the primers, the capped primers in there, and that those then can go on to prime viral mRNA synthesis. In the discussion, the authors say, we've noticed that this N protein can substitute for EIF4F. They don't show the data. I have a feeling they saw the other paper or heard of the other data. And so they say, we know this also. And they say this means the following, or this suggests the following mechanism. N protein is bound to the capped primer in the P-body. At some point it comes out, it primes mRNA synthesis. The N protein is bound to it the whole time. When the mRNA is finished being made, then the N protein can participate in translation by the mechanism that we talked about in the previous paper, substituting for EIF4F. Did I say influenza virus this whole time? I did.
Yes, you. I think you said that a couple of times.
I'm gonna have to fix this. So this is also hantavirus, which is a bunyavirus.
Search and replace.
Oh,
yes.
Cool. It was a tough one to do with audio, though. Now we correct ourselves. So there are 3 viral families whose members prime mRNA synthesis with capped primers. orthomyxoviruses, the flu viruses, bunyaviruses, which include the hantas, and the arenaviruses like
And I was
Lassa.
focusing so much on this mechanism because
Yeah.
I hadn't seen these papers before today. So typical of when I would show up unprepared for advanced topics class back in grad school. I'm trying to read ahead in the paper. I'm like, oh, wait, at the cap. Oh, the Peabody's. Right. I remember Peabody's.
I remember those days. I would call on students, and they would
Yes.
be. I asked them a question, and they'd be reading the paper,
Flipping frantically
through, trying to find. I remember one student of mine who said, "Eric, tell me you don't know. I don't want to wait till you read the bloody paper in front of the class." Anyway, yes. So both of these papers have to do with hantavirus and protein. These viruses all need primers, capped primers derived from the cell, and so the hantavirus N substitutes for EIF4F. It also binds the capped primers that hantaviruses use and stores them in P-bodies. A reasonable question is whether all of this happens with influenza virus as well.
And I realized why you were talking about influenza a moment ago, because that's the next thing we're going to discuss.
That's right. We're going to talk about the endonuclease. Now, the cool thing is, well, the question here, of course, is whether translation of hantavirus mRNAs is independent of EIF4F. So this whole model that the authors propose, which we just reviewed, that the N protein not only is there when the cap primers prime mRNA synthesis, but it remains there for translation. They propose that for hantaviruses. That model implies that translation of these viral RNAs is independent of cap binding protein. And that's actually been shown before. Curiously, and here's where I was going with all this, influenza mRNA translation also does not seem to require EIF4E, the cap binding protein. Okay.
Right.
And if N is substituting for that, that would make perfect sense.
Exactly.
So the question now is whether flu primers are stored in P-bodies or not. And that hasn't been looked at. I'm sure someone's doing it now because flu mRNA synthesis is primed with cap oligonucleotides. So a likely scenario is that those are also stored in P-bodies, and maybe the viral NP protein is binding to them.
Right. If one virus figured out how to do this, others probably did too.
Yep. Viruses are very okay. So that is a hantavirus. So yes, now these primers are made when the viruses encode endonucleases that cleave cellular mRNAs to produce these primers. And there was a very nice paper this week, Advance Online in Nature, called The Cap-snatching Endonuclease of Influenza Virus Polymerase Resides in the PA Subunit. Now, previously, we thought that the endonuclease of influenza virus that cleaves the mRNAs of the cell to produce these primers was in two other subunits of the polymerase. They're either PB1 or PB2. So basically, this very nice paper, it's a combination of biochemistry and structural biology. They show that the endonuclease is actually in the PA subunit, which is not what people thought at all previously. And that's very interesting. They express a piece of this protein, and they show that it has the requisite activity. They also have a structure of the protein, and it looks just like an endonuclease, so it all makes perfect sense. And the nice thing now is that we know exactly where the endonuclease is, and you can use the expressed protein to screen for new antivirals.
Right.
And because if you don't know exactly what protein the activity is in, it's difficult to do a drug screen.
Right. Drug developers like to have a specific target.
This endonuclease is actually present in the virus particle, but I think it would be very difficult to develop a drug screening assay with it. I don't think there's very much, and it would be rather unreliable to set up such a screen, right? So having the protein expressed is much better. So this is good because we can really go ahead now and look for antivirals that target this. And as we've said before, we need as many as we can get for influenza virus.
And targeting as many different life cycle stages as possible.
Exactly. Which actually brings something up. There was some discussion on my virology blog this past week. I'm not sure if we discussed it on this podcast, but certainly on my blog I discussed all the new influenza virus inhibitors that are being developed. And there's some new neuraminidase inhibitors, and there's some new targets like polymerase.
Yeah, I think we talked about this last episode or two episodes.
Yeah. So one of these new influenza neuraminidase inhibitors is called peramivir, which I think is in phase 3 now. And the papers on this drug show that, in fact, the mutations that cause resistance of H1N1 strains to oseltamivir also cause resistance to this new drug.
Right.
So someone wrote, what's the Why are they bothering? Which is a good point. And probably the answer is that they started years ago before any of the resistance emerged.
Exactly.
And someone actually commented that we could have predicted that the resistance would emerge. Pretty much for any antiviral, yeah, you could say that resistance is going to emerge and you would be a good predictor, right?
Right, but saying the resistance is going to emerge doesn't really help you in preventing it. And you have to develop the drugs because, they'll be useful until the resistance emerges, and in the case of flu, it just happened a lot faster than people had expected.
It could still be, even though this year many of the circulating H1N1 strains are resistant, who knows what kind of selective forces will be imposed on the virus next season.
Exactly.
It could result in the circulation of a totally different strain. Now, I was told in the discussions that I was an idiot for proposing this, basically. But in knowing what I know about viruses, I don't think it's totally impossible.
Well, and in the case of flu, we're not talking about something that's endemic. It doesn't continue circulating in the population very much during the summer. What we get is a fresh batch. More or less fresh batch every year, which is why we have to keep changing the vaccine.
Yeah, at least in temperate climates, it doesn't go.
Yes, right.
This company will have to make a difficult decision, right, whether to market this or not.
Exactly.
But of course, it's also still effective against H3N2 strains. Okay, now I know this year those aren't the major strains circulating, but again, who knows? It could change, right?
Yep.
You got to be a little, what should we say, sanguine when you talk about resistance to antivirals.
Yeah, and you have to just not throw stuff away quickly. There are lots and lots of bacteria that are resistant to penicillin, but we still need to manufacture penicillin because there are a lot of others that aren't.
Exactly. Yeah. All right. Another interesting paper that I noticed in the past week was about a cellular protein called tetherin. And I had not heard of this before, but a paper came out on Ebola, and this paper was in PNAS. Tetherin-mediated restriction of filovirus budding is antagonized by the Ebola glycoprotein. So tetherin is this very interesting protein in the cell. It's in the plasma membrane and other places as well. Previously, it was shown to inhibit the release of HIV virions from the cell. So apparently the virions bud on the surface. The budding is complete, but maybe they're still attached by this protein called tetherin, and it holds it there. And so basically it reduces the release of infectious viruses from the cell. So it's pretty neat.
Yeah.
So this tetherin is actually a protein induced by interferon. So early in infection, when cells sense a virus infection, they make interferon, and then this is one of the hundreds of genes that are induced to try and set up an antiviral state. And so makes perfect sense to try and target virus release from the cell.
It's your last chance.
Yeah. And there are all the ISGs, all the interferon-stimulated genes probably target the whole continuum of viral replication, starting from entry to translation, RNA synthesis, assembly, and release.
Right.
We've only begun to scratch the surface of understanding that. This one, for HIV, what's cool is that the VPU protein of HIV, one of these small proteins, antagonizes tetherin. So if you take VPU out of HIV, the yield in the presence of tetherin is very low. But if you put VPU back in, apparently VPU antagonizes tetherin, and the virus can grow in the presence of the protein. Now, in this paper, they showed the equivalent for Ebola virus. Okay, they showed that tetherin antagonizes the release of Ebola from cells. And that interestingly, the Ebola glycoprotein G can antagonize the mechanism or the effect of tetherin. So that's very cool. We don't know how this works. We don't know how tetherin really works. We don't know how the viral proteins antagonize it, but we know the effect.
Well, and the other interesting thing is they closed the loop, too. They found that Ebola glycoprotein can substitute for HIV VPU. So if you take the Ebola glycoprotein, apparently, and add it to a culture of HIV-1 that lacks VPU, you can boost the yields back up again in the presence of tetherin.
That's right. Very good. That's really cool experiment. Yeah. So it's a pretty universal mechanism, apparently.
Right. They seem to be working the same way.
So I wonder if other envelope viruses are also affected by tetherin. Influenza, the flaviviruses, the togas, the alphas. So I'm sure that all the labs will be looking at this.
Or if they're not affected by tetherin, how do they avoid it?
So this is good. I like this. This is very cool.
Yeah.
Neat stuff. Before we continue talking about viruses, here's a brief message from the American Society for Microbiology.
On May 17th through the 21st at the Pennsylvania Convention Center in Philadelphia, the American Society for Microbiology will hold its 109th General Meeting, the largest annual gathering of microbiologists in the world. Visit the General Meeting website at gm.asm.org to view the preliminary program, register for the meeting, or reserve your hotel stay. That's gm.asm.org.
The last thing we'll talk about is a letter, Emerging Infectious Diseases. And this was a letter from a couple of scientists. The title of the letter is Time from Illness Onset to Death, 1918 Influenza and Pneumococcal Pneumonia. Basically what they're saying is, as we all know, in 1918, this influenza pandemic was very lethal. It killed a lot of people. This is why we're worried about the next pandemic, because we're afraid that it's going to be a similar virus, very lethal. So we're all making these antivirals left and right. But these guys say they think that, in fact, because there weren't any antibiotics then, that in fact it was strep pneumonia that was killing most of the people. And they look at the mean time to death here in the 1918 outbreak, and they say it was basically too long to be viral pneumonia. And so they say we should be focusing on antibiotics as well as antivirals, but don't forget the antibiotics, that may be a big component of it. I heard this theory years ago. When I was in graduate school, people said this. I don't know why it's coming up again now. Maybe they're trying to temper this antiviral steamroller that's going or something. But the 1918 virus has been reconstructed, as you know, from the sequence.
Right.
It's very lethal in mice and in ferrets. So it does have properties in lab animals that are consistent with it being very virulent in people. So I don't know the story here. I emailed Peter Palazzi, who's an expert on flu and who happened to have been my thesis advisor, so he'll listen to my emails. And he hasn't answered me yet. I wanted to know what he thought of this, and if he gets back by the next podcast, we'll talk about it. But I don't know. I'm just, it may be an issue, but I'm sure it's not the only part of the lethality. I'm sure the virus also contributed. What do you think, Alan?
Yeah, this kind of idea has been kicking around, not just for the 1918 flu, but for other historical plagues. The classic example, of course, being the Black Death, which killed a third of Europe in the 13th century. And, the problem with these historical events is that, in the case of the Black Death, we have no pathology samples whatsoever. We have absolutely no way of knowing what happened except from the written record. And it's pretty clear when you look back at that and you look at the symptoms that there were multiple diseases going on. Everybody blames bubonic plague, but in fact, there were superimposed problems going on. The 1918 flu is very similar. In that case, we have some pathology samples, but if you look at the reports, you see that probably what was happening was not just a pandemic of a single virus. There was that. But layered on top of that, you had a war going on, and people are going to be getting sick as a result of that, and they're going to be getting sick from a wide variety of things, and you had no antibiotics. And so it's virtually certain that some of the people who are calculated among the millions of deaths for 1918 flu actually did die of bacterial pneumonia, because that was a disease that occurred and that was a disease that killed people. And if everybody else is dying from influenza, then it's all going to be called influenza. So there is certainly, the waters are certainly muddier than you might believe if you were just reading the news reports. And also, it's worth noting when the virus was reconstructed a few years ago, one of the challenges in reconstructing it, they went back to pathology slides. I think pathology slides from Walter Reed from that collection, and they had to find one where they had good samples that were from a patient who did not have any bacterial pneumonia complicating the case. And apparently that was a bit of a challenge, because that's a common secondary problem. So again, this comes up. Well, how many of these patients were actually dying of strep pneumonia, for example, and how many were really dying of the virus? And of course, ultimately, we can't know. But as you say, it's very telling that when the virus was finally reconstructed and put into laboratory animals that are normally pretty good predictors of these things, it does seem to be highly pathogenic. So I think it's good to have these kinds of letters and people reminding folks that that story is not quite as clean as you might think. And yes, we should absolutely be worried a bit about 1918 flu, and we should be worried a bit about bacterial pneumonia.
I agree. You don't want to focus on one thing. It was multifactorial, as we say. So, and it would be interesting to know what Mark Crislip thinks of this. Mark, if you're listening, weigh in. Mark of the Infectious Disease Puscast.
Yes.
All right. Now we have some emails to share with you, and two of them deal with the last episode of TWiV, I believe. Everett wrote, Regarding the rabies patient, I heard Dr. Despommier mention a teen who had recovered from rabies. I'm not too sure if this is the case that he was talking about, but to my understanding, this is the first case of a patient who had symptomatic rabies with no prior prophylaxis before onset of symptoms. So Dick thought that there might be a recent survival of rabies, but there in fact has only been one, and Everett sent us some links, and he is right. There was a young woman in Wisconsin some time ago who is apparently the only individual to survive rabies without having been vaccinated, and she was placed in a coma by her physicians, which was probably why she survived, and I don't know if Dick was referring to that or not. I think Kaprowski, when he talked at Columbia in 2005, probably was referring to this case. He had said she was severely compromised, but apparently this young lady has gone to college, and she's pretty good. She's fine, so she's had a remarkable recovery.
Yeah.
Some other people have survived rabies, but they were all immunized, so their vaccine didn't take very well, and they maybe had moderate disease and so forth. So these are vaccine failures, and you can find those, and we'll put some links up. Now, as for who Dick was thinking about, in fact, there was a young lady in the UK who acquired rabies just in December, and she died.
Right.
And she contracted rabies while working in Africa, and she was from Northern Ireland. She was in Africa, and two dogs were fighting, and she tried to separate them, and she got scratched or bitten, and she came back to the UK, developed rabies, and died. She didn't survive, unfortunately. That's an example. It's really lethal. Very difficult to survive after a rabies bite. So thanks, Everett, for correcting that. That's great. And in fact, I heard about that in my virology meeting in Switzerland that I went a few weeks ago. They talked about this case because they were focusing on importation of diseases from other areas, and that fit the bill. Okay, then another one from Vanny, who said, hi guys, I really enjoy the podcast. It's a great way to learn about cool papers that I probably wouldn't have come across any other way. Just wanted to comment on the last episode, Seminal Discoveries. I think Saul mentioned there were two vaccines. These are HPV vaccines, a trivalent and quadrivalent. However, there is a bivalent and a quadrivalent. So he's correcting Saul. The bivalent is for HPV 16 and 18, which are the high risk for carcinoma. And the quadrivalent has 16 and 18, as well as 6 and 11, which are causing genital warts. So he writes, which is very good. I want to correct that mistake because I think it's important for girls to get the correct information about this great vaccine. So thanks a lot. Saul said, you're absolutely right. He had a brain freeze and forgot. Mark Crislip, actually, of the Infectious Disease Puscast. So we had made his podcast a pick of the week some time ago, and last time we picked his science blog, and he wrote back, thank you, you are too kind. Even my ravenous ego is sated. P.S. I have started listening to TWiV and quite enjoy it. That's great, Mark. Well, his podcast is wonderful. I really enjoy it. He does it all by himself, Alan. You can laugh because he's a funny guy.
He actually manages to do stand-up infectious disease.
By himself. Yeah, it's really good. So if you're listening, Mark, it's great. Please think about the flu pneumonia issue.
Yes.
So this brings us to our picks of the week. I have a great time picking these things. Science blog of the week. Mystery Rays from Outer Space is the name of the blog. This is written by an assistant professor of immunology and virology at Michigan State University, and he works on viral immunity. And he talks about viruses, immunology, and stuff on his podcast. Now, what does the title come from? He says, for years, Mystery Rays from Outer Space has been my explanation for anything that seems inexplicable, which covers much of immunology and virology. Well,
yes.
That's a cool title. And I have to tell a story. I was once being an expert witness for a vaccine case, and I was saying how in virology, sometimes you get one result one day and a different result another day. And the lawyer said, well, why is that, Dr. Racaniello? And I said, I don't know. Sometimes it's the phase of the moon. And he got all bent out of shape when I said that. I said, never mind. I took it. Strike it from the record, please. So it's the same thing as mystery rays. Anyway, whoever you are, this fellow doesn't identify himself. He just says he's an assistant professor. You shouldn't hide. People want to know who you are, and he doesn't answer his emails either. I've commented and emailed him, so you need to be a little more open. That's how you will get more people. Anyway, it's a good blog. Check it out. Science podcast of the week. So the Emerging Infectious Disease Journal that we've talked about a number of times from CDC, they have a podcast, which is pretty short, about 8 minutes. It just highlights key articles. This week is about Creutzfeldt-Jakob, for example. So they talk to an expert about various things. It's pretty good. And finally, this is up Alan's alley, our science book of the week, The Coming Plague by Laurie Garrett. You know this book, Alan.
Oh, yeah.
It's about newly emerging diseases in a world out of balance. It's actually about 1995, right?
I think that's right. This was right after, either right after or right before she did Betrayal of Trust, which was a somewhat longer book about the global status of global public health.
So she was a journalist for Newsday on Long Island, correct?
Yes, and I don't know if she still is.
And so this book, The Coming Plague, is basically talking about all the emerging infections that are hitting us and where they come from and why. So if you've been at all interested in this issue, which we talk about all the time because it's one of the fascinating aspects of virology, you should read this book. It's quite big, but it's well done.
She doesn't tell short stories, but the interesting thing is she's very old school journalism. So she'll go places and do things that you just cringe, thinking, "Oh my God, you're there!" And one of I forget which of these books it was in. She goes to the heart of an Ebola outbreak, and she's there reporting on the outbreak from the place and going and interviewing people and like standing near patients who are bleeding out from this disease, and it's gripping reading. It's certainly not something that's upbeat. You may need something a little lighter afterward, but it's very, very interesting stuff.
So, Alan, you're saying that you wouldn't go to an Ebola outbreak to cover it?
I don't think that I would.
I don't blame you.
And it wouldn't just be the Ebola that I would be concerned about.
I have a Laurie Garrett story. She used to, anyway, teach a course at Columbia down on the Morningside campus. And one of my former graduate students took this course. And she came back one day and she said, Garrett is calling the RNAs of influenza virus chromosomes. You keep saying the influenza virus chromosomes. Is that right? And I said, absolutely wrong. I mean, it's not the right terminology. A chromosome has a specific meaning in biology. It's DNA and protein chromatin. And so she went back and told Garrett, and she said, Garrett just said, it doesn't matter. Wouldn't give in, basically. So she's just stubborn. Using the right terminology. I think in science, you have to make sure what you're saying is correct. Otherwise, people are going to not understand. If you say chromosomes, talking about what mammals and other organisms have in them, and it ain't what flu has.
That's right.
So that's Laurie Garrett.
But it's worth saying her reporting is generally quite good.
Yes. Well, she's a reporter, so perhaps she shouldn't be teaching a class about viruses. I don't know.
Right.
Yeah, we all have our strengths. Just a few things to wrap it up. There's an interesting new documentary that just has come out. You might want to check out. It's called Naturally Obsessed, The Making of a Scientist. And this was actually filmed in labs at Columbia by the Rifkins. Richard and Susan Rifkin, yeah. I think. Have you seen this, Alan?
I have not, but I totally want to. And I didn't see, I was just looking at the website and I didn't see that there was an easy way to see the film.
No, they had a screening at Columbia a few weeks ago for us. I didn't see it. I think you need to go somewhere and see it and actually pay money.
Right.
It's not. They haven't put it online for free.
No, I would pay money to see it, but in my case, I'd probably have to order a DVD or something, and that's clearly not an option.
But it may be out there. Check it out. It's pretty cool. So I am still playing around on Twitter. I find it useful. My handle is ProfVRR. Alan is Alan Dove, one word. Check us out there. Our website is TWiV.tv, and you can send us email at twiv@twiv.tv if you want to ask us questions or correct things that we say or suggest topics for future shows. I have a whole list of great topics for future shows that I've received. Some of them include how vaccines are made, bacteriophages, plant viruses. I'm working on getting that together, so send in your suggestions. Anything else from you, Alan?
No.
You're content?
I'm quite happy.
Very good. You've been listening to This Week in Virology, where we try to make virology interesting. Thanks for joining us. We will be back next week. I don't know who will be back. I will be back. Maybe Alan, maybe Dick, maybe a guest host. Check in and see. Another TWiV is viral.